geneticist. Thank you. Working in very diverse tropical and temperate forest ecosystems. Katrin did her master and PhD degrees at University of Ulm, Germany. And then she moved to Marburg for 2 consecutive postdocs. And recently, she got a professorship position at University of Freiburg to build up a new forest genetics group. And yes, we are very lucky to have you here. Thank you very much for coming. And we look forward to see your talk. Please, the floor is yours.
Thank you, Ben, for the invitation. And I'm very happy to be here. I've been here a couple of times, and I always enjoy being with the colleagues here, being at the WSL. So today I'll introduce you to some of my research. And just when we talked earlier, I realized there's not a single bit of European forests in there today, unfortunately. So I hope you will not be too disappointed. There are temperate forests, but not European temperate forests. But I do a lot of research also within European forests because it's way easier to work out of the door. But today it will be more remote what I'm going to present. So, like many of you, I'm fascinated by forest ecosystems, no matter whether it's temperate ecosystems or super species-rich tropical ecosystems. I was always fascinated by how all these organisms living there interact in different ways and form this ecosystems that somehow work and are disrupted and recover. And of course now, one of the main topics that I'm also interested in is this enormous threat that forests are confronted with because of all the increasing climatic extreme events, pest outbreaks. Also in the tropics, there's a lot of fragmentation, land use change going on. And of course, with our forest management, we also have an impact how forests might look in the future. So my main research topics or goals are to unravel the molecular basis of adaptation and acclimation in forest trees, because this is very important to understand how they will perform, especially now that we are confronted with a very uncertain future. And I'm interested in what causes spatial patterns of genetic and epigenetic diversity, and what are the consequences of this spatial distribution. Depending on the specific research questions, I work at different life stages. So most of my projects are actually with adult plants, but I also work a lot on seed dispersal and recruitment, reestablishment, and depending on the question, also working with seeds or seedlings or saplings might be the target. So today, I'm going to introduce you to 3 of my research topics, which are first the genetic basis of adaptive traits and local adaptation. Then we'll move to tropical habitats, where I currently have a big project on phenology of tropical tree species. And then I go back a bit to where I'm coming from. So I started out as an ecologist and was investigating mostly seed dispersal. So we will have a quick look on how dispersal influences genetic diversity and gene flow in the tropics. And a last chapter I'm not presenting here probably because of time. We might discuss later on, but let's see. So that's epigenetics, which is a whole new exciting field, but also very complicated to do in trees. So let's start. So you know probably this triangle. Christian and his colleagues have probably talked in previous talks a lot about it. So if you want to understand how trees cope in their environment, we have to simultaneously look at phenotype, environment, and genotype. And one of the approaches I've been following up for a couple of years by now, and we also work together with Ben and Christian and Patrick and others here from the WSL on that, is the joint analysis of dendroecological and genetic data. So one of the main questions here is, do trees that cope better under past climatic conditions share specific alleles? And today I want to present one study that was recently published by Melanie Zacharias. She defended her PhD just a couple of weeks ago, and nicely the day before her defense, she got her main article accepted in molecular ecology, which was very nice. She worked in Alaska. So unfortunately, I haven't been there myself, but it looks very, very interesting. And there she worked in Picea glauca in white spruce, and investigated tree line and forest stand population, and wanted to know whether the individual reaction to drought stress differs between different types of tree lines. So she worked at cold and drought limited tree lines, and also between the tree line and the forest population. And we wanted to also see whether in these different sites that we investigated, whether they have similar or different signatures of adaptation. So her her sites were located in Brooks Range and Alaska Range, which are both cold limited tree lines, whereas interior Alaska is more drought limited tree line. And in all of these locations, we had a total of about 500 trees. So we selected them. So we selected only the adult trees that had a minimum DBH of at least 10 centimeters, and thus an age of at least 50 years. So we had a long or sufficiently long tree ring series there. And we sampled at each site the tree line and the forest sites. And because of certain restrictions, it was not very even, but yeah, we thought we can handle that design. What we then did is we looked at the tree ring chronologies. So you see here the tree ring index growth over the years. And then we identified pointer years, so years where most of the trees seem to have suffered, seem to have a growth decline. So let's see. So this pointer has a bit of a lag. Let's see. Yeah. It feels a bit like you are drunk when you point with this. So we identified these years, and then among all the years where they had kind of disjoint growth decline, we looked into the climatic data to identify those where, did I do that? Okay. Where they had negative indexes for water availability. So we really just wanted to identify those years where the trees really suffered from drought and not from any other kind of limitation. So for most of the sites, we only found 1 or 2 of these years. And then we genotyped these trees with about 3,000 SNPs and then ran association analysis. So we associated the growth in these years of the individual trees, the growth performance, with these SNPs, and followed one single SNP approach in Genesis, and the other was a multiple SNP approach using a Bayesian sparse linear mixed model. And there we closely collaborate with Ben. So if you have specific questions on these methods, you might get some more sophisticated answers here in the audience. And with these approaches, we could identify some genes that were obviously significantly associated with these respective growth performance. So for the single SNP association, we only found genes when we jointly analyzed. So we did this analysis individually for each of the sites, but also when they had a common drought year, we also joined them, which might, of course, be then, and we discussed this very intensively in the paper, also be correlated to the fact that they're different populations, but also within individual sites, we found a number of genes with the multi-SNP association that were linked. With the 2 approaches, we also had 2 genes that were identified by both approaches, which supports a bit that they're indeed related to this growth performance. And the interesting thing is that when we looked into the annotation of the genes, is that really some of them were linked to for example, genes that are relevant for the membranes or for enzymes that are involved in the growth. And also, we had a very intensive look at other studies in white spruce. There has been quite some work done. And indeed, a quite significant part of the genes that we identified were also associated to wood anatomy in common garden trials, for example, and were differentially expressed under drought. So we are quite positive that these genes are indeed involved in this growth regulation. The interesting thing, however, was that we really found different genes in each population. So it seems that it's not, that is really local adaptation going on if there's a signature, and that it's not kind of a joint reaction if there's more drought or a real limitation then it's just disspecific genes where the trees can react. So that means there's not the one answer the tree can give, but there are several answers because, of course, there's a lot of genes and that are involved in these growth reactions, and you can either use that one or that one to hopefully then perform better on the drought. A bit of a similar project we have been running in Patagonia. So here, Jill Seckley is the PhD student, and here we were sampling in the Patagonian Andes. This project is with Notophagus pumilio, a conifer species that has a very interesting distribution range. So the green part here is the distribution range. So it has a very long north-south distribution along the Andes. And we thought this is a very cool sampling design to look for local adaptation because, of course, there's this extreme day length gradient. And of course, if you consider circadian clock regulation, you might expect that there's some kind of adaptation to learn at what light conditions do I actually resume growth, for example, in spring and so on. And the other interesting part is that coming from the coast, there is a huge precipitation gradient. So at the Chilean coast, you have 4,000 or 5,000 milliliters of precipitation, and the distribution range goes down to, I think it's like 400 or 500 millimeters per year. So it's really extreme. As you can already see, we collaborated with Argentinian partners, so all our sampling sites are on the Argentinian side, so we don't cover the whole precipitation gradient, unfortunately. But you still see, especially these red dots are a bit kind of the sampling sites that are already into the more drier areas. Yeah, and you can already see, if you compare these pictures, so you have these very erect growth, so they form huge trees with huge stands, but depending on where they grow, they have also on the tree line, this very crippled growth. And you can see here, that's a very xeric site that is really in extreme environments that they are exposed to. So we sampled in 20 sites in total, and we copied a bit the sampling design that we had in the Gentry project, in the European project, where you also collaborated. So we had 20 populations in a paired design, always sampling high and low elevations. And we then carried out an exome capture where we used the same genes that were used for the Fagus study in Europe. So one of the not yet finalized, but planned analysis is then to compare what we found in terms of adaptation with these European and Patagonian species. Unfortunately, when you then do all this genotyping and you filter rather stringently, there's not so many SNPs left after all these filtering SNPs, but we still ended up with about 13,000 SNPs. So these are meant to be in the other order. So yeah, that's the sampling sites again. So in total, it's about 500 trees in these 20 sampling sites. And so we had a first look at the population structure, and you can see that there's a pretty clear divide, more or less in the middle, into two big clusters. And then you can also see the most northern population. So the colors in the structure plots stand for genetic clusters, and depending on how many groups you divide, you see step by step which clusters are more closely in terms of genetics to each other. And you can see on top, the most northern population is behaving a bit differently, in a way so differently that for some analysis we excluded it later on, and we somehow suspect that there might even be some hybridization going on, but we are not entirely sure. But for the association analysis, we excluded that population. What we found when we looked at genetic diversity is a very interesting north-south gradient. So no matter on what measure for genetic diversity we looked, whether it's allelic richness, nucleotide diversity, or private allele count, they are always more diverse in the northern population compared to the southern population, which is already a really interesting result. And then we used several methods to look for signatures of local adaptation. So first, we just looked at signatures of selection solely based on the genetic data with PCA, adapt, and bypass. And we also associated it to environmental variables with bypass and LFMM. And the most interesting part for me here is that there's 41 genes or alleles that are identified by all of these approaches. So there's for sure something going on here. And the other result that we already have is that most of these genes that have a signature of selection are linked to temperature and not so much to precipitation. And also there's a significant number, so the middle part is the average day length in January. So there seems also, as we had expected, something going on with these extreme day lengths temperature. We are not yet fully done with this analysis, so I can only show you one of these summaries of the annotation, which, well, we see water deprivation here, so that might also direct show us some direction that there is something still going on with drought, but that's about where we are, and Jill will of course continue this analysis and we'll have more finite results hopefully in the very near future. Nevertheless, we can already say that there are very distinct genetic clusters. There's a very distinct diversity gradient, north-south orientated, and at least in the part that we investigated, the temperature seems to exert a stronger selection pressure. However, as I said, we didn't cover the whole part where there's a lot of precipitation, and that would be, of course, a very interesting investigation for the future to really then get a collaboration also with Chilean partners, so we can really look into this whole gradient of very high to very low precipitation. So now we move a bit north, but actually north in that case means to more warmer habitats, to Ecuador. And there I have an ongoing project on phenology of tropical tree species. So of course here in temperate zones, it's a very well-known and very familiar phenomenon that you have phenological events like this mass flowering of cherry in Japan at certain times of the year. And this is particularly interesting because we can see that in recent years, the flowering happens earlier and earlier. So under climate change conditions, phenology is one of the things in nature that are really affected directly, and where you can really observe this effect. What is less known, however, that also in tropical regions, there's a lot of tree species that flower at certain times of a year, that produce new leaves at certain times of the year in the absence of real seasons. So especially in the parts where I'm working in evergreen tropical rainforests, there might be differences, of course, over the year in terms of precipitation, but especially in Ecuador, where we are really at the equator, there's no daylight difference. There's really almost no difference in temperature. So how do these trees actually know from the weather or climatic conditions when it's time and to synchronize? And they need to synchronize, not so much, of course, because they might freeze otherwise, but because many of the tree species growing there, they are very rare. And so to reproduce successfully, they need mating partners. Or, for example, other organisms that interact with them are also seasonal. And then they try to produce new leaves, for example, at times of the year when there are less insects, to avoid having all their new leaves eaten. So it's very important for them to synchronize. There is a selection pressure, but it's not so much the seasons, but it's more interactions within and among species that force them to be synchronous and timed. But what do they use? And there's a lot of theories, but no one really knows whether it's light conditions or precipitation or whatever. Some even claimed for a while that they can sense these some minutes of daylight differences, which I doubt, but you never know. So, and phenology also, of course, then influences, for example, the reflectance of the canopy. It influences how carbon sequestration is distributed over the year, if they produce new leaves and lose leaves and so on. So there's also a feedback to the atmosphere. And finally, as I already said, the selection pressure actually might be these plant-animal interactions. And of course, at the same time, if the trees are then synchronous and timed, they also influence again how species interact with them. And so in this project, we have two big goals. We want to try and decipher what are they actually reacting to. And we want to see how does that influence interactions. And specifically, we're going to look at herbivory and pollination. So how does the timing and degree of synchrony in the species affects the efficiency of pollination, for example? So we work in southern Ecuador. The station we work at is located at about 2,000 meters. It's quite a challenging terrain. You can see it's very steep. And since we select samples there in the trees and have cameras placed in the trees, we sample buds and so on, we have to climb. And that means being with all these climbing equipment in this terrain, and you see how steep it is really quite a challenge. And for example, to access our sampling sites every day, we have to cross the river, and that's our bridge. So it's quite exciting to work there and quite hard. We work on 4 species there. 2 of them are deciduous, so Handranthus and Cedrella montana. They both shed their leaves at certain times of the year and produce new leaves. And Handranthus is our main species because you can see when they flower, they're really, really beautiful. And they occur at 2,000 meters, but they also grow specifically in dry forests. So we have a second site in the dry forest where we also work with them. There is, of course, a clear seasonal structure, and they are much more synchronized there. And we also want to compare the regulation there. And Graphenrida imaginata and Alsatea verticillata, they also have a synchronous and more or less timed flowering. So we knew that from previous studies. And we chose those species because we specifically want to look into first monitoring with a very high resolution leaf and flowering phenology. So we have cameras positioned in all of the trees. So we really have a daily resolution, because most of the studies in the past were done by direct observations. And then of course you are limited and you can only visit them so often. So we hope we really have a daily resolution here. And then we will look into molecular phenology, which means that we will, of course, what you observe when you look at them is there's a flowering peak. But of course, that's not when the tree is actually reacting to an environmental cue. It reacts much earlier. And the first way where we can see now the tree is actually reacting to something is by the expression of certain genes. So that's why we go into the trees, take these samples out, and then we will conduct gene expression analysis to pinpoint a time point when the trees start reacting to environmental cues. And thereby, we hope that we can get much closer to deciphering what are they actually reacting to. And then, as I said, we are interested in the interaction with herbivores and pollinators. And since they are not totally synchronized, there's kind of a peak, but you also have earlier and later individuals. And there then we are, for example, interested whether the diversity of pollen donors differs. There should be less fathers if you are early and late. And maybe this has an influence in the diversity of the emerging flowers, of the emerging offspring. It might have an effect on the actual seed set, and maybe also on the performance of the seedling. So that's another avenue we are looking into here. And yeah, with the herbivores, we are also interested in at which age of the leaves and at which seasons during the life cycle of the leaves is herbivory most intense. Yeah, that's part of our team in Ecuador. That was during our climbing course that we did there in January. And yeah, it's a lot of fun, but it's also a lot of tough field work. So sometimes my PhD students are also suffering a bit. But at the same time, they're always like, but it's so cool here. So they have good and bad times there. Yeah, and with that, I'm already at my last chapter, and this is going back a bit. So I started my career out in the University of Ulm as an ecologist, and I worked there in a group that studied bats, and I really wanted to work with that professor, but he studied bats, and I'm not as night active. And I was interested in all these interactions. So we found somehow a compromise, and we decided that I'll work on dispersal by bats. So my first project was actually on dispersal of fig fruits and fig seeds, of course, by frugivorous bats. And then later I entered a collaboration with primatologists, and that's what I'm talking about now. So the approaches there were basically very simple. So the idea is that the further seeds are dispersed, the more diverse a population is. So we have here on the y-axis a measure for genetic relatedness over geographic distance. And depending on how limited seed dispersal is, the more closely related the individuals are that grow close together. But if you have very far seed dispersal, you wouldn't expect any such pattern in the populations. So I collaborated with Tiziana Gernicandusio, who was a PhD student at the time at the German Primate Center, and one of her work part of her work was a review, and she looked into all those studies that investigated spatial genetic structure in animal dispersed species. And indeed, we found the pattern that in these highly diverse frugivores that you see in the first box plot, spatial genetic structure is very, very low. Whereas when you have less, what we classified as less mobile frugivores, so smaller animals that move not so far, you have a higher structure. Also, when you have roosting species or scatter hoarders at short distances, you have a higher spatial genetic structure. So there is more a signature of relatedness compared to long distance hoarders, for example. So there seems something to being on which is related to the foraging behavior. And this study was carried out in Peru. So this station is located just 70 kilometers from Iquitos. But you can only reach it by boat. So it's also a quite nice trip of 1.5 days to get to the station. And then you usually take food and everything for 3 to 4 weeks and just stay at this isolated station without any internet connection or such to the rest of the world. And the cool thing is that they're habituated tamarins. So they're just so used to the researchers that you can approach them and observe them very closely. And they have long-term data on these tamarins, and they have not so big home ranges. You can see here, the scale is just 1,000 meters, and these are 3 different groups over several years, and they really stay in their home range. And they also know a lot about the species they are eating, and so we focused on 2 tree species that are, to our knowledge, exclusively dispersed by these monkeys. Parkia panorensis is a large canopy tree with a rather low density. It produces a quite high number of fruit pods with seeds that are surrounded by a gum that is eaten by the monkeys. And the other tree species is Leonia cymosa. It's a small understory tree, very small trees, just with a higher density, but only a few ripe fruits at a given time. So we investigated the dispersal of these fruits by the monkeys in 1 or 2 groups. So, and also collected all the adult trees we knew, additionally saplings, and they did behavioral observations. So each time they saw them eating a fruit and defecating, they tried to recover the fecal samples. So we could also do a matching with genetics. So from the observational data, we saw that in the first graph, you see the number of simultaneous feeder in any given tree. If they feed in Leonia, it's only 1 or 2 of the monkeys that can actually enter the tree at the same time. Whereas while they feed in Parkia, it's often the entire group being in the same tree. And this also affects how fast they travel afterwards. Because when they all are in the same tree and they get saturated, they do not travel as far and they rather stay together. So it really influences at least a bit how they behave afterwards, and also when they decide to rest again. So they rest earlier when they feed in the same tree in Parkia compared to when they feed in Leonia. And then we use different approaches to estimate dispersal distances. So these are again the 2 trees, Parkia and Leonia. The first observation is the direct dispersal. Then we have the genetic matching of the dispersed seeds. The third box plot is with seedlings, and then 2 modeling approaches where we use the behavior and the gut passage time to model how far could these seeds dispersed if they remain within the tamarine for a certain amount of time, and we knew how they traveled. And with almost all of the approaches, we found that the seed dispersal distances are longer in Leonia cymosa, except for the one approach where we looked into seedlings, but there we might actually also have included a number of seeds that just fell to the ground. So that might bias these results a bit. And with the genetic data, we investigated then in seedlings, juveniles, and adult trees, the spatial genetic structure. And we found that this is much more pronounced in Parkia, the big canopy tree, where we observed the shorter dispersal distances. So this all somehow comes together. So indeed, more individuals were feeding in the big canopy tree in Parkia versus Leonia. So the distance to the next resting site was also shorter. And as a result, dispersal distances were shorter, and we have a more pronounced spatial genetic structure as a consequence in Parkia versus Leonia. Unfortunately, this station is not funded anymore, but I will hopefully now continue my collaboration nevertheless with the primatologists. So we have now Malika as a PhD student, and she's now going to Ecuador, to the Chocó region, which is one of the super biodiversity hotspots of this world. And she will work with a highly threatened primate species, the brown-headed spider monkey. And this monkey feeds on selectively locked tree species. So in some of these areas, they are extracted. So the abundance of these tree species is significantly reduced and they are more disturbed. And we suppose that this will have a big effect on the behavior, foraging behavior of these monkeys. And we want to see how all of this, the changed foraging behavior and also the extraction of a significant part of the adult individuals affects then the genetic diversity of the regenerating seedlings of these species. So I'm looking forward to this project in the next coming years. Because from all what we've learned, we are convinced that the behavior of dispersers really determines the distribution of genetic diversity, and much more research should be done here as well. So there was the last chapter. We decided to not talk about it now, but who's interested in epigenetics in trees can join and come to Christian's and Ben's office at 1, and then we can have a little chat about epigenetics in trees. Also, we are looking currently for postdoc for an epigenetic project. If you know someone, let us know. Project first 2 years in Marburg and then 1 year in Freiburg. And yeah, I hope I could show you that all of this research across the entire life cycle of trees in different types of habitats is super interesting and fascinating. There's, of course, a whole lot of people who contributed. And I want to really thank all my collaborators, students, and of course you for your attention.
Great talk. Thank you very much, Katrin. Is there any question in the audience? Yes, we have a few minutes. Katrin.
I'll ask a question I know nothing about. I really enjoyed your talk, but so I feel like I'm not really up to date at all on all of the genetic stuff, but I always wonder when people focus on genes, like that's the final, or that's maybe the smallest scale, but it could be also that there's something at the level of the pathways.
Yeah.
And then I always wonder if it's the pathway or the gene that's important and how you know, it's a bit like the scaling of, I don't know how you would say it, but of genetic process or something like this. And I just, it's more of a question, is there a pattern at this? Is that a true thing that this other scale is important? And if so, have you looked at it, or
so
how do you look at it?
The level of regulation?
Yeah, so like these genes, instead of, it could be that all the genes are within a single pathway, but they're different genes, but they're still somehow constrained by being part of the regulatory pathway, if I understand this at all.
Yeah, I mean, there are different levels to that. Of course, to reach, for example, a certain goal, to be more resistant to drought, it could be either that pathway or that pathway or that pathway. And that's why many of these, it's so hard to disentangle which genes are really responsible, because it's often not one gene, not two genes, but often hundreds of genes that might be involved. And of course, there might be different solutions. That's what I try to show a bit. One population might find a solution by changing in these 100 alleles, and another might be these 500 alleles where there are slight changes and differences. And then, of course, on top of that, there might be this post-regulation part. So that's the epigenetic part, for example, because the DNA can be more or less accessible, and that changes then gene expression. So not the gene or the protein per se, but the abundance of the protein. And that can, of course, also have a phenotypic effect. And that's why it's so complex and so hard to understand and to really pinpoint what's going on. And that's why we often lack these clear answers that people want to have from the genetics, because it's such an interesting puzzle. And I mean, there are these studies on human height. You would assume that if height in humans is a very highly heritable trait, that we should by now understand what's going on there. But it's hundreds, thousands of genes that are important for just that one trait. And then it's probably the same for tree growth, because there's so many pathways involved, of course, in determining the final growth at the end of the year of a tree. It's super hard to pinpoint, but we have to start somewhere.
Thanks for the very interesting presentation. I was interested in single SNP association and multi-SNP association methods that you use, and I was wondering if you use both of them to probably I missed the exact reason, to compare the filtering, like the result of the filtering approach of the genes, and in a different case, why would you use only the single SNP association method or the multi-SNP association
I mean,
approach?
all of these methods that are around have their advantages and disadvantages. And of course, also all our data sets have their advantages and disadvantages. And so one general approach is that we use several of these methods. I guess you can, of course, add to that, Ben. What's your point of view there? But we use several methods, and then if we identify SNPs with several methods, we feel more confident that this is a real positive association.
But which, so I was wondering, in which case would you prefer to use the single SNP association or the multi-SNP? Like, what would you expect from one instead of the other approach? I don't know.
I mean, that's a bit, they're different, again, different aspects. So of course, if you had like, by coincidence, that one gene that is super important, you would hopefully find this with the single SNP association. So, but often it's the interaction between many genes, and then you would have to look at them simultaneously. There is not the golden road. Ben, do you want to add to that?
So those different programs rely on different statistics. So they have also assumptions. So you have to rely on those. And I would say a combination, a multiple evidence is always good. So yeah, I would then go back to the annotation and see if there are interesting gene pathways involved in what you would expect. But otherwise, experimental work is, of course, the must. Yeah.
Yeah. I mean, that's, of course, the tricky thing. We want to see this pattern in natural populations, but these environments are so uncontrolled that you can never really characterize the environment a single tree is experiencing. So you never know if maybe the root of the one tree has access to some leftover water in the ground, whereas the other has not. You don't never see that. And that's why it's so complex, and that, yeah, we are trying to decipher that, but that's why the next joint project we are planning is taking place in highly controlled environments with a clone. So we have very limited genetic diversity, very restricted environments. But I think both approaches have their value, and we should not focus only on one, but we have to be very aware of the respective limitations.
Aaron.
Thank you. So it was really great talk. I have a question on the slide that you presented the diversity across the elevational gradient. And you said that the diversity is higher at the higher altitudes?
No, in the north.
To northern tree line. So if you could kindly go to that slide.
So maybe let's have a quick look at the map first to make that clear. So what we have, so here, of course, since we are in Argentina, the warmer populations are in the north. And so there it's rather high elevation populations. And then the further south we get, the colder it gets. And they're actually not so high. So they're almost at sea level down here. Okay. So just to get the geographics right. And then these populations in the north that are in higher elevations,
I see.
They have the higher genetic diversity, whereas those that are really at the point where it's very harsh conditions, very short summers, they have a lower genetic diversity.
Yeah, thank you. So my question is, like, when we say high elevation or the northern population, it means they are probably in the warmer condition.
Yeah.
And maybe because of climate change, they're getting more and more warmer, perhaps. So when it is better to grow in a certain condition, normally it's the selection could take reduce the diversity, or like in general, like that's what I thought, like in under stress or under very good condition is kind of less diverse. I mean, how you interpret this high diversity and the in this condition? I mean, what we understood from this high diversity?
I mean, out front, I have to, of course, admit that I'm much more familiar with the glacial history here in Europe than in Argentina. That's why we closely collaborate with the Argentinian partners, and they do that part of the discussion. But of course, you would assume first that during ice ages and so on, the surviving populations would have been located further north. So that's where you would assume that refugia are. Plus, I would assume a much stronger selection pressure in those northern, more extreme populations. And I think a combination of that could indeed lead to a reduction of genetic diversity. But for an in-depth discussion on that, I would have to refer to my Argentinian colleagues.
Thanks, Ben. Just building on this, you mentioned that the most northern population is a putative hybrid population. Do you see signals of introgression in terms of different alleles that are present in that population that taper off throughout the distribution or anything like that?
Yeah, you know, there's always the things that you regret after you have done the genotyping. And in that case, our regret was that we didn't include samples of the sister species.
Okay.
So I can't tell you. They are different, but we don't know whether this different is linked to the potential sister species. I can't say, unfortunately. I hope I can tell you at one point in the future.
Okay, thanks.
Okay, this question is from Mario Pesendorfer, and he asks, Nothofagus pumilio is a strongly masting species. Do you see any interactions between seed production and short-term drought adaptations?
So I don't have the data, of course. So the species, I mean, even here we have masting observations for very limited populations and so on. And I think there's more interest now here in that because, of course, there's also more masting events, but I can't answer that. We have dendro data from this plot, so we did the same as in the other projects that we got the dendro course. So we have the growth data, so we can potentially say something about drought adaptation at one point, and also link then the drought reaction to the genes, but we don't have the link to the masting. So we don't have that data.
Thank you. And then there's one more question from Elizabeth Martinez, who asks, or says, thanks for your talk. Unfortunately, I could not be there. Question about Alaska. Is drought the main driver of tree ring series in tree line in Alaska? I suspect that not. If so, how sure are you that the real limitations are not overriding tree growth when you look at drought effects?
So, as I said, two of these sites are rather cold limited and drought limited, but nevertheless, there has been a long-standing study also, especially of tree ring data on these sites. And according to our colleagues that collaborated there with the tree ring analysis, Martin Wilmking from Greifswald, they told us that yes, it's one of the limiting factors. Of course, we could have looked into other things, but that was at least one that we could clearly try to pinpoint with the respective drought years. So of course, there are other limits. And as we have discussed in length, it's super hard to really tell the ultimate reason why a year caused a better or a worse growth in the past. But since they already have some experience in these plots, I guess that it's not totally far-fetched to assume that at least in these years, drought was one of the really serious limits.
Last question.
Am I really the last question? Okay. So I was interested in your cameras and the canopy. And so then I guess it's more of a methodological question of, is there some sort of automated way that you're getting the data on the phenology from there? And then you hinted that you were actually trying to understand the environmental cues of this phenology, but we didn't get an answer about that. So I guess that's still work in progress.
Yeah.
But then the question is do you have sort of, or what is the scale of the temperature or precipitation measurements that you're taking? Are they right there in the same tree, or are they more of a regional scale? I guess it's hard to know what scale those cues are acting at, maybe.
Yeah, I mean, why I choose the site is that they have a really long time series of high resolution meteorological measurements. So there's a meteorological, or there are geologists and geographers working there, and they have all the measurements on site. So it's not on the tree, but really in the site where we have the measurements from. Otherwise, that would have been a far-fetched, of course, because especially precipitation can be very regional. I mean, some of our trees might be 500 meters away from the meteorological station, but that's the scale. And for the cameras, that's local cameras, because of course we can't connect them to anything. So as we go there regularly to the trees to collect samples anyhow, we just retrieve data manually. I think that was.
Yeah, but then how do you go? Then you have I guess you have a lot of images, and then are those automated, or is it still manual looking to see, it's just 7 flowers?
There are some, so we try to run them through some R packages that help us selecting foggy from less foggy pictures, but in the end, we still have to look at a lot of pictures. I mean, we try to do it half automated, but it's often not as satisfactory, and then you still have kind of a look. And we started the project this year in February, and we are planning to collect data for 3 years.
Okay.
So that's why no results are there yet, but I thought it's nevertheless interesting to tell what's going on. But yeah, the main results will only be available if we have several years of information, of course.
Okay, thanks.
There is still one question. Okay, there is time.
Yeah, thank you, Katrin. I have a question for your last study. I mean, if you are a tree species that is only dispersed by this monkey, and you're actually only dispersed by one group, so as I understood, because they don't move. So that's the worst case, no? You cannot disperse. How do you do that? Are there like occasional long distance dispersal events, or?
That's what we assume. I mean, of course, these groups, they are rather constant, and that's why we chose it, because we thought it's a very neat system. We would have expected a much stronger structure, but of course you have gene flow via pollen, and that we cannot track in the same way. So the dispersal is indeed limited, but of course there is some individual sometimes escaping or going somewhere else. So the family structure is rather stable, but of course not entirely, because of course also the tamarines need to mix. So I guess there are occasional events.
I have a question. As a follow-up project for the tree phenology initiative, do you think it would be possible to integrate these biotic factors as predictor and then do this kind of landscape genomics approach where we look at significant associations between interactions, biotic interactions, and genetic variants?
You mean with the tree species we study in Ecuador?
Yes.
I mean, the problem is, when I first thought about the project and what could I do in these terms, I of course thought about kind of a landscape thing, but the problem with these tree species is that they have such a low density. So one reason for choosing these 4 species was that they're in the site where we work, they have a rather high abundance. So we could identify a couple of individuals, but it's so hard to find these individuals depending on where you go. So Graphenrieder, for example, is fairly common, so you could probably do that, but to be honest, that's not the most interesting of the 4 species. With Chondroanthus, you usually know where they are because you can find them when they're flowering. So we are really thinking about going landscape at a later time point, but it's not so straightforward because you have interesting distributions and abundances most of all. So to find a certain number of trees is often not straightforward. And I mean, with the gene expression, we will really focus on a certain set of genes. So if there's something that might be a really interesting pathway, especially because Chondroanthus, the yellow flowering tree, it, for example, in the high mountains, it flowers in August, September, and in the dry forest, it flowers in January. So there's for sure something interesting going on. So once we've understood that a bit more, we it would be very cool to have follow-up studies on there. But yeah, that's the problem with this project. Oh, we should include that and that aspect. But yeah, I mean, you have to limit it to something and collect good data for certain questions first.
Cool, thanks.
My question involved like I know this is not related to any of your research focuses, but since you talked about molecular phenology, I just was curious to know if you're also looking at secondary metabolites produced in the trees, whether they are given stress, so some tree species might produce more of a protective metabolite to protect themselves compared to other ones. I just wanted to know if you're doing research over that.
Of course, that's one of the things that comes up constantly, and yeah, my PhD students are struggling a bit. It's a lot of field work, and they constantly have new ideas, but we should also sample that and that and that and that. And of course, it would be super cool to also have time series of metabolite production over the years. So we will actually now start sampling some trees. We don't know yet when and how to analyze that, because of course we have not yet funds for that, and you need someone to do it. But yes, of course, that's one of the questions that pop up immediately here.
Katrin, you have a last question? Katrin has an announcement.
Okay. Before we just close up, or maybe just after we close up, and I cannot see that. I just want to announce the next BD seminar will be Laura Pollock, and that will be on October 22nd. And she comes from North America or Canada and has done really, really cool stuff with conservation planning and many other things. Was a postdoc with Wilfred Touillet for many years and is now a professor. So that would be probably pretty broad and super cool. And then we also have a Biodiversity Symposium on December 6th. And the idea is there we have 3 really high impact provocative speakers, we think, and the idea is to have a cool symposium and then a bit of a party and discussion so that people can start to think about the Biodiversity Center more and what it has to offer, etc. So it's a bit of we could never have a party to start the center because it was in the middle of COVID. So this is a bit of our, not official starting party, but sort of a party nonetheless, because it's now possible and straightforward. And so I hope that you will all attend this and continue to come to these seminars, and even also propose some for the spring. Thanks. Oh yes, thank you. And now we're starting the journal club again. Oh my goodness. And that's tomorrow. The first paper will be actually on AI, machine learning stuff. It's a really cool review of a group that's very well known in that field. But this journal club works because people come and participate. So it's super fun. It used to be before COVID, a really dynamic discussion. And we are starting that up, and that will be twice a month. And after we get the schedule sorted out, and we'll have it right before beer once, and then another 2 weeks later. So it'll be twice a month. One of them, not currently because the scheduling is hard, but one will be right before beer. So you drink a beer and then keep discussing, or discuss and then drink a beer. And then, yeah. Anyway, so just keep tuned for those. But this Thursday is the first one. So you're very welcome. At 3:30. Thank you. Oh, I'm sorry, the 26th, not the 22nd. Thank you. Okay.
Thank you very much, Katrin, again for your talk. And for those interested, we have lunch together, so please join and have fun. Ciao.